Hey there all viewers. Welcome back to another Healthy Lifestyle Supports Channel's video. In our latest video, we already talked about the 11 best foods that boost brain health and memory. Also, we promised to talk more about the 7 worst foods for your brain and memory within this video. Actually, your brain is the most important organ in your body. It plays important roles to keep your heart beating, lungs breathing and all the systems in your body functioning. That's why it's essential to keep your brain working in optimum condition with a healthy diet. Eating the right foods to keep your brain healthy can dramatically decrease your risk of developing neurological problems later in life. However, some foods have negative effects on the brain, impacting your memory and mood and increasing your risk of dementia. Estimates predict that dementia will affect more than 65 million people worldwide by 2030. Luckily, you can help reduce your risk of the disease by cutting certain foods out of your diet. Well, this video reveals the 7 worst foods for your brain and memory that you should limit or avoid, as following. Number 1. Sugary drinks. Sugary drinks include beverages like soda, sports drinks, energy drinks and fruit juice. A high intake of sugary drinks not only expands your waistline and boosts your risk of type 2 diabetes and heart disease, it also has a negative effect on your brain. An excessive intake of sugary drinks increases the odds of developing type 2 diabetes, which has been shown to increase the risk of Alzheimer's disease. In addition, higher sugar levels in the blood can increase the risk of dementia, even in people without diabetes. A primary component of many sugary drinks is high fructose corn syrup, HFCS, which consists of 55% fructose and 45% glucose. A high intake of fructose can lead to obesity, high blood pressure, high blood fats, diabetes and arterial dysfunction. These aspects of metabolic syndrome may lead to an increase in the long-term risk of developing dementia. Animal studies have shown that a high fructose intake can lead to insulin resistance in the brain, as well as a reduction in brain function, memory, learning and the formation of brain neurons. One study in rats found that a diet high in sugar increased brain inflammation and impaired memory. Additionally, rats that consumed a diet consisting of 11% high fructose corn syrup were worse than those whose diets consisted of 11% regular sugar. Another study found that rats fed a high fructose diet gained more weight, had worse blood sugar control and a higher risk of metabolic disorders and memory impairments. While further studies in humans are needed, the results suggest that a high intake of fructose from sugary drinks may have additional negative effects on the brain, beyond the effects of sugar. Some alternatives to sugary drinks include water, unsweetened iced tea, vegetable juice and unsweetened dairy products. Number 2. Refined carbs. Refined carbohydrates include sugars and highly processed grains, such as white flour. These types of carbs generally have a high glycemic index, GI. This means your body digests them quickly, causing a spike in your blood sugar and insulin levels. Also, when eaten in larger quantities, these foods often have a high glycemic load, GL. The glycemic load refers to how much a food raises your blood sugar levels, based on the serving size. Foods that are high glycemic index and high glycemic load have been found to impair brain function. Research has shown that just a single meal with a high glycemic load can impair memory in both children and adults. Another study in healthy university students found that those who had a higher intake of fat and refined sugar also had poorer memory. This effect on memory may be due to inflammation of the hippocampus, a part of the brain that affects some aspects of memory, as well as responsiveness to hunger and fullness cues. Inflammation is recognized as a risk factor for degenerative diseases of the brain, including Alzheimer's disease and dementia. For example, one study looked at elderly people who consumed more than 58% of their daily calories in the form of carbohydrates. The study found they had almost double the risk of mild mental impairment and dementia. Carbohydrates may have other effects on the brain too. For example, one study found that children aged 6 to 7 who consumed diets high in refined carbs also scored lower on nonverbal intelligence. However, this study could not determine whether consuming refined carbs caused these lower scores, or simply whether the two factors were related. Healthy, lower glycemic index carbs include foods such as vegetables, fruits, legumes and whole grains. You can use this linked database to find the glycemic index and glycemic load of common foods. Number 3. Foods high in trans fats. Trans fats are a type of unsaturated fat that can have a detrimental effect on brain health. While trans fats occur naturally in animal products like meat and dairy, these are not a major concern. It's industrially produced trans fats, also known as hydrogenated vegetable oils, that are a problem. These artificial trans fats can be found in shortening, margarine, frosting, snack foods, ready-made cakes and prepackaged cookies. Studies have found that, when people consume higher amounts of trans fats, they tend to have an increased risk of Alzheimer's disease, poorer memory, lower brain volume and cognitive decline. However, some studies have not found an association between trans fat intake and brain health. Nonetheless, trans fats should be avoided. They have a negative effect on many other aspects of health, including heart health and inflammation. The evidence on saturated fat is mixed. 3 observational studies have found a positive association between saturated fat intake and the risk of Alzheimer's disease, whereas a 4th study showed the opposite effect. One cause for this may be that a subset of the test populations had a genetic susceptibility to the disease, which is caused by a gene known as APOE4. However, more research is required on this topic. One study of 38 women found that those who consumed more saturated fat relative to unsaturated fat performed worse on memory and recognition measures. Thus, it may be that the relative ratios of fat in the diet are an important factor, not just a type of fat itself. For example, diets high in omega-3 fatty acids have been found to help protect against cognitive decline. Omega-3s increase the secretion of anti-inflammatory compounds in the brain and can have a protective effect, especially in older adults. You can increase the amount of omega-3 fats in your diet by eating foods like fish, chia seeds, flax seeds and walnuts. Number 4. Highly processed foods. Highly processed foods tend to be high in sugar, added fats and salt. They include foods such as chips, sweets, instant noodles, microwave popcorn, store-bought sauces and ready-made meals. These foods are usually high in calories and low in other nutrients. They're exactly the kinds of foods that cause weight gain, which can have a negative effect on your brain health. A study in 243 people found increased fat around the organs, or visceral fat, is associated with brain tissue damage. Another study in 130 people found there's a measurable decrease in brain tissue even in the early stages of metabolic syndrome. The nutrient composition of processed foods in the Western diet can also negatively affect the brain and contribute to the development of degenerative diseases. A study including 52 people found that a diet high in unhealthy ingredients resulted in lower levels of sugar metabolism in the brain and a decrease in brain tissue. These factors are thought to be markers for Alzheimer's disease. Another study including 18,080 people found that a diet high in fried foods and processed meats is associated with lower scores in learning and memory. Similar results were found in another large-scale study in 5,038 people. A diet high in red meat, processed meat, baked beans and fried food was associated with inflammation and a faster decline in reasoning over 10 years. In animal studies, rats fed a high-fat, high-sugar diet for 8 months showed impaired learning ability and negative changes to brain plasticity. Another study found that rats fed a high-calorie diet experienced disruptions to the blood-brain barrier. The blood-brain barrier is a membrane between the brain and blood supply for the rest of the body. It helps protect the brain by preventing some substances from entering. One of the ways processed foods may negatively impact the brain is by reducing the production of a molecule called brain-derived neurotrophic factor, BDNF. This molecule is found in various parts of the brain, including the hippocampus, and it's important for long-term memory, learning and the growth of new neurons. Therefore, any reduction can have negative impacts on these functions. You can avoid processed foods by eating mostly fresh, whole foods such as fruits, vegetables, nuts, seeds, legumes, meat and fish. Additionally, a Mediterranean-style diet has been shown to protect against cognitive decline. Number 5. Aspartame. Aspartame is an artificial sweetener used in many sugar-free products. People often choose to use it when trying to lose weight or avoid sugar when they have diabetes. It is also found in many commercial products not specifically targeted at people with diabetes. However, this widely used sweetener has also been linked to behavioral and cognitive problems, though the research has been controversial. Aspartame is made of phenylalanine, methanol and aspartic acid. Phenylalanine can cross the blood-brain barrier and might disrupt the production of neurotransmitters. Additionally, aspartame is a chemical stressor and may increase the brain's vulnerability to oxidative stress. Some scientists have suggested these factors may cause negative effects on learning and emotions, which have been observed when aspartame is consumed in excess. One study looked at the effects of a high aspartame diet. Participants consumed about 11 milligrams of aspartame for every pound of their body weight, 25 milligrams per kilograms, for 8 days. By the end of the study, they were more irritable, had a higher rate of depression, and performed worse on mental tests. Another study found people who consumed artificially sweetened soft drinks had an increased risk of stroke and dementia, though the exact type of sweetener was not specified. Some experimental research in mice and rats has also supported these findings. A study of repeated aspartame intake in mice found that it impaired memory and increased oxidative stress in the brain. Another found that long-term intake led to an imbalance in antioxidant status in the brain. Other animal experiments have not found any negative effects, though these were often large, single-dose experiments, rather than long-term ones. Additionally, mice and rats are reportedly 60 times less sensitive to phenylalanine than humans. Despite these findings, aspartame is still considered to be a safe sweetener overall, if people consume it at about 18 to 23 milligrams per pound, 40 to 50 milligrams per kilograms, of body weight per day or less. According to these guidelines, a 150-pound, 68-kilogram person should keep their aspartame intake under about 3,400 milligrams per day, at the maximum. For reference, a packet of sweetener contains about 35 milligrams of aspartame, and a regular 12-ounce, 340 milliliter can of diet soda contains about 180 milligrams. Amounts may vary depending on brand. In addition, a number of papers have reported that aspartame has no adverse effects. However, if you prefer to avoid it, you could simply cut artificial sweeteners and excess sugar from your diet altogether. Number 6. Alcohol. When consumed in moderation, alcohol can be an enjoyable addition to a nice meal. However, excessive consumption can have serious effects on the brain. Chronic alcohol use results in a reduction in brain volume, metabolic changes and disruption of neurotransmitters, which are chemicals the brain uses to communicate. People with alcoholism often have a deficiency in vitamin B1. This can lead to a brain disorder called Wernicke's encephalopathy, which in turn can develop into Korsakoff syndrome. This syndrome is distinguished by severe damage to the brain, including memory loss, disturbances in eyesight, confusion, and unsteadiness. Excessive consumption of alcohol can also have negative effects in non-alcoholics. Heavy one-off drinking episodes are known as binge drinking. These acute episodes can cause the brain to interpret emotional cues differently than normal. For example, people have a reduced sensitivity to sad faces and an increased sensitivity to angry faces. It's thought that these changes to emotion recognition may be a cause of alcohol-related aggression. Furthermore, alcohol consumption during pregnancy can have devastating effects on the fetus. Given that its brain is still developing, the toxic effects of alcohol can result in developmental disorders like fetal alcohol syndrome. The effect of alcohol abuse in teenagers can also be particularly damaging, as the brain is still developing. Teenagers who drink alcohol have abnormalities in brain structure, function and behavior, compared to those who don't. Particularly, alcoholic beverages mixed with energy drinks are concerning. They result in increased rates of binge drinking, impaired driving, risky behavior and an increased risk of alcohol dependence. An additional effect of alcohol is the disruption of sleep patterns. Drinking a large amount of alcohol before bed is associated with poor sleep quality, which can lead to chronic sleep deprivation. However, moderate alcohol consumption may have beneficial effects, including improved heart health and a reduced risk of diabetes. These beneficial effects have been particularly noted in moderate wine consumption of 1 glass per day. Overall, you should avoid excessive alcohol consumption, especially if you're a teenager or young adult, and avoid binge drinking entirely. If you are pregnant, it is safest to avoid drinking alcohol altogether. Number 7. Fish high in mercury. Mercury is a heavy metal contaminant and neurological poison that can be stored for a long time in animal tissues. Long-lived, predatory fish are particularly susceptible to accumulating mercury and can carry amounts over 1 million times the concentration of their surrounding water. For this reason, the primary food source of mercury in humans is seafood, particularly wild varieties. After a person ingests mercury, it spreads all around their body, concentrating in the brain, liver and kidneys. In pregnant women, it also concentrates in the placenta and fetus. The effects of mercury toxicity include disruption of the central nervous system and neurotransmitters and stimulation of neurotoxins, resulting in damage to the brain. For developing fetuses and young children, mercury can disrupt brain development and cause the destruction of cell components. This can lead to cerebral palsy and other developmental delays and deficits. However, most fish are not a significant source of mercury. In fact, it is a high-quality protein and contains many important nutrients, such as omega-3s, vitamin B12, zinc, iron and magnesium. Therefore, it is important to include fish as part of a healthy diet. Generally, it is recommended that adults eat 2 to 3 servings of fish per week. However, if you're eating shark or swordfish, only consume 1 serving, and then no other fish that week. Pregnant women and children should avoid or limit high mercury fish, including shark, swordfish, tuna, orange roughy, king mackerel and tilefish. However, it's still safe to have 2 to 3 servings of other low mercury fish per week. Recommendations may differ from country to country, depending on the types of fish in your area, so it's always best to check with your local food safety agency for the recommendations that are right for you. Also, if you are catching your own fish, it is a good idea to check with local authorities about the levels of mercury in the water you are fishing from. Summary: your diet definitely has a big impact on your brain health. Inflammatory diet patterns that are high in sugar, refined carbs, unhealthy fats and processed foods can contribute to impaired memory and learning, as well as increase your risk of diseases such as Alzheimer's and dementia. Several other substances in food are dangerous for your brain too. Alcohol can cause massive damage to the brain when consumed in large quantities, while mercury found in seafood can be neurotoxic and permanently damage developing brains. However, this doesn't mean you must avoid all these foods completely. In fact, some foods like alcohol and fish also have health benefits. One of the best things you can do for your brain is to follow a diet rich in healthy, fresh whole foods impact brain health. Well, that's all now. What have you learned from this video? Please tell us in the comment below. Please hit like and subscribe this channel in order to ensure that you won't miss any videos. Thanks for watching this video from the beginning till end. Please eat safe, stay safe, and live together. Don't forget to take care of your brain.